Today we are going to talk about cellular respiration. The first step of cellular respiration is glycolysis, which is the breakdown of glucose in the cytoplasm of the cell. This step occurs in the cytoplasm because it is an anaerobic reaction, therefore it does not need oxygen. The remaining steps are aerobic, as they require oxygen and occur in the mitochondria. As a general overview, you can see that glucose will eventually be broken down into pyruvate and ATP. Glycolysis begins with glucose, or C6H12O6, which is drawn here as a 6-carbon chain. 2 ATPs are then used to break the glucose into 2 3-carbon chains and add a phosphate group to each chain. This phosphate group comes from the ATP molecules used earlier. These chains are called pGal molecules. Each pGal molecule then produces one pyruvate molecule. Each pGal to pyruvate reaction also results in 2 ATPs and one NADH molecule. Overall, the breakdown of one glucose molecule with 2 ATPs results in 2 pyruvate, 4 ATP molecules, and 2 NADH. However, the NADH is not entirely produced by this reaction. Instead, an NADH plus molecule is reduced, so a hydrogen atom is added to the molecule, therefore resulting in an NADH molecule. Because 2 ATPs were used in the beginning, the net reaction yields 2 ATPs, 2 NADH molecules, which will later be used in the electron transport chain, and 2 pyruvate molecules, which will also later be used as acetyl-CoA in the Krebs cycle.
At the end of glycolysis, 2 pyruvate molecules still contain a large amount of extractable energy. The process of extracting the remaining energy in the form of ATP from the 2 pyruvate molecules is known as pyruvate oxidation. In eukaryotic cells, this process takes place in the matrix, the innermost part of the mitochondria. In prokaryotic cells, this process takes place in the cytoplasm. Pyruvate oxidation converts pyruvate, a 3-carbon molecule, into acetyl-CoA, a 2-carbon molecule attached to coenzyme A, producing an NADH and releasing 1 carbon dioxide molecule in the process. Acetyl-CoA is a product used to fuel the Krebs cycle in the next stage of the cellular respiration process.
Hello everyone, and today I'll be teaching you about the citric acid cycle. The citric acid cycle goes by many names, including the Krebs cycle or the TCA cycle. It was originally named after German scientist Hans Krebs. Here is a full diagram of the Krebs cycle. Each step of the cycle is catalyzed by a specific enzyme which creates specific products. I know it looks really complicated here, but I'm going to be breaking it down for you step by step. Before we start talking about the steps of the Krebs cycle, we have to talk about where it occurs. The Krebs cycle occurs in the mitochondria, specifically in the matrix of the mitochondria. The matrix is a gel-like substance inside the second membrane of the organelle. It's much like the cytoplasm inside of a cell, except it's less viscous and contains less water. The first step of the Krebs cycle is pyruvate oxidation, which we've already talked about, so this should be a great review. A 3-carbon pyruvate molecule breaks down into a 2-carbon acetyl-CoA molecule. The CoA at the end of acetyl-CoA is short for coenzyme A. Then, another organic molecule comes along by the name of oxalacetate, which is a 4-carbon molecule. Basic math time. When a 4-carbon molecule reacts with a 2-carbon molecule, what do you get? A 6-carbon molecule, of course. The 6-carbon molecule is called citrate. The coenzyme A is kicked out of this reaction, but we'll be seeing more of it later. The next step of the cycle involves the citrate produced by the former reaction. The citrate becomes isocitrate, a very similar molecule. Isocitrate still has 6 carbons, much like citrate. Then, isocitrate breaks down into a 5-carbon molecule known as alpha-ketoglutarate. You don't have to remember the name of this molecule, but just note that it has 5 carbons. But what happened to the 6th carbon? It didn't just magically disappear. The 1 carbon that is lost is released as carbon dioxide, which has 1 carbon, as you should know. In this step, a molecule called NAD+ is produced. Spoiler alert, NAD+ will become a molecule called NADH, which is a coenzyme. Step 4. The 5-carbon molecule called alpha-ketoglutarate from the last reaction becomes a 4-carbon molecule called succinyl-CoA. There's that coenzyme A from earlier once again. 1 carbon is released as carbon dioxide and another NAD+ molecule is produced. This step is very similar to the last step of the cycle. Note that the same products were produced, with the exception of the number of carbons involved. As I mentioned earlier, the 2 NAD+ molecules become 2 NADH molecules. But how? Reduction. A reduction reaction is basically a reaction that results in a gain of electrons. The NAD+ has a +1 positive charge, and 1 electron has a -1 negative charge. When the two bonds, the charges cancel out each other. As you may know, an element with 1 proton and 1 electron is hydrogen. That's where the H in NADH comes from. Alright, back to the main action. As you remember, a product from the last reaction was succinyl-CoA. In this step, the coenzyme A gets swapped out for a phosphate group. Then, this phosphate group transfers once more to an adenosine diphosphate molecule, which becomes an adenosine triphosphate molecule, also known as ATP. This is the only time ATP is produced in the entire cycle. The 4-carbon molecule remaining is a molecule called succinate. The 4-carbon molecule succinate oxidizes, which means it loses electrons. In this case, 2 hydrogen atoms are lost from the succinate molecule. The 4-carbon molecule remaining is called fumarate. These 2 hydrogen atoms don't just disappear, they're used to reduce FAD, a cofactor, into FADH2. Next, the fumarate molecule reacts with water to form a 4-carbon molecule called malate. The 4-carbon molecule called malate oxidizes, meaning it loses an electron, and an NAD+ gains this electron and becomes NADH. The oxidation of the malate molecule causes the malate to become oxalacetate, and no, you're not experiencing déjà vu. Oxalacetate was the molecule from the first step which reacted with pyruvate to make citrate. So we start all over again. It's a cycle. So there it all is, the Krebs cycle. You may have noticed there weren't many ATPs produced in this cycle at all. That's because the FADH2 molecule and 3 NADH molecules created from one rotation of the cycle go off to the electron transport chain to act as electron carriers. This is where the majority of ATP is produced. So from the top, glucose creates 2 pyruvate molecules, and 2 pyruvate molecules yield 2 acetyl-CoA molecules. One turn of the Krebs cycle requires 1 acetyl-CoA molecule, so 1 glucose yields 2 turns of the whole cycle.
So now we'll be talking about oxidative phosphorylation. And within that, we're going to talk about the electron transport chain and chemiosmosis. So the first step of oxidative phosphorylation is the electron transport chain. It's a chain of proteins and molecules found in the inner membrane of the mitochondria. The proteins are organized into 4 large complexes labeled 1 to 4. The first two complexes receive electrons from NADH and FADH2. When the electrons are transferred to molecules near the beginning of the chain, the carriers revert back to NAD plus and FAD, while the H plus is added into the matrix. NADH transfers its electrons directly to the first complex. However, FADH2 isn't as good at donating electrons. It gives its electrons to complex 2, which doesn't pump protons across the membrane like the first complex does. As electrons are passed down the chain, they move from higher energy levels to lower energy levels, and the released energy is used to pump H plus ions, or protons, out of the matrix. This pumping establishes an electrochemical gradient. The last complex passes the oxygens to O2, which splits into 2 oxygen atoms and accepts protons from the matrix to form water. And that leads us to the second part of oxidative phosphorylation, chemiosmosis.
So the last step of oxidative phosphorylation is chemiosmosis. Chemiosmosis is the movement of hydrogen ions down their electrochemical gradient in order to form ATP. Due to pumps in the electron transport chain, the concentration of H plus is greater in the intermembrane space than it is in the matrix. To correct this, H plus ions move through a channel protein called ATP synthase back into the matrix. As H plus ions go down their electrochemical gradient and move down the protein, it catalyzes the addition of a phosphate to ADP in order to make ATP. The energy that you get from ATP comes from this proton gradient. The end of oxidative phosphorylation yields 30 to 32 ATP.
So what happens if you don't have oxygen in order to carry out oxidative phosphorylation? In that case, the cells undergo anaerobic respiration. Specifically, we'll be talking about fermentation. Now you have 2 kinds of fermentation: lactic acid fermentation and alcohol fermentation. Lactic acid fermentation results in lactate, which is the deprotonated form of lactic acid. In this process, NADH transfers its electrons directly to pyruvate. It donates its electron to pyruvate, and in that process, it loses the hydrogen, and that way it can be reused in the process again. The other fermentation process is alcohol fermentation. In alcohol fermentation, the NADH donates its electrons to a derivative of pyruvate and produces ethanol. In this case, it donates its protons to 2-acetaldehyde in order to produce 2-ethanol. In this process of fermentation, the NADH donates its electrons, loses the hydrogen, and that way NAD plus can be reused in the process again and produce energy. Both cases end up producing 2 ATP altogether.